Simple animals. After completing the laboratory session, you should be able to describe the distinguishing characteristics of the taxonomic group studied and their evolutionary advances. Identify representatives of the group studied. Identify and state the function of the structural components of the animal studied, and define all terms in bold print. In this week's lab, we will investigate simple animals. We are members of the kingdom Animalia, but the animals we encounter in lab are about as different from humans as animals can get. Let's first consider our similarities. All animals are multicellular, and these cells contain membrane-bound organelles. This is what separates eukaryotes from prokaryotes. Animals are therefore members of the domain Eukarya. All animals are heterotrophs who cannot produce organic molecules from the sun's energy, but rather ingest plant or animal products. Finally, all animals are capable of moving, and they do so using several forms of contractile fibers. Most animals engage in sexual reproduction, specifically gametic sexual reproduction. Mature individuals exist in diploid form, i.e., they have two sets of chromosomal information. In order to engage in sexual reproduction, animals produce specialized sex cells called gametes that, through the process of meiosis, contain only one set of chromosomal information. These sex cells are haploid. In males, testes produce gametes called sperm, which are typically much smaller than female sex cells called eggs. At fertilization, sperm and egg unite to produce a zygote, which matures into a diploid organism. Now that we have addressed their similarities, let's consider how we classify animals according to their differences. Symmetry is one important characteristic, with some animals exhibiting radial symmetry, some exhibiting bilateral symmetry, and some exhibiting neither radial nor bilateral symmetry. Some animals exhibit radial symmetry. When they are divided along any plane, the two halves are symmetrical. Bilaterally symmetrical organisms, as the name implies, can be divided along the mid-sagittal plane, the longitudinal axis of the body, and both halves will be similar. Of the organisms we will investigate today, sponges are asymmetrical or radially symmetrical. Cnidarians exhibit bilateral symmetry, and flatworms and roundworms exhibit bilateral symmetry. Another important characteristic used to classify animals regards their level of organization. Animals have either tissue, organ, or organ system levels of organization. Organisms with radial symmetry have tissues only, while bilaterally symmetrical animals have organs or organ systems. Radial symmetry is an older evolutionary development from which bilateral symmetry evolved. Bilaterally symmetrical organisms can exhibit three morphologies related to their embryonic tissues, which are also called germ layers. And these embryonic tissues are also related to the resulting body cavity called the coelom. These layers result in a body plan that is either sac-like or tube-like. The tube can be unlined or contain a lining. Let's look at these in more depth. Acoelomates, for example, flatworms, lack a coelom. The contents of their internal organization are adjacent to the body wall and also adjacent to their gut. There is no true cavity. Cnidarians that have only tissue-level organization are also sac-like. However, sponges have no germ layers and therefore they have no body cavity or body plan at all. Pseudocoelomates, like roundworms, have a fluid-filled space between the mesoderm, the middle layer, and the endoderm, i.e. the gut, the inner layer. Most organs are in the fluid-filled cavity. Coelomates, like animals, have a body plan whose components are all lined with mesoderm-derived tissue. We will not investigate any coelomates in this lab, but you can conceive of both pseudocoelomates and coelomates as having a tube within a tube body design. In embryonic form, coelomates can develop in two ways. They can be protostomes or deuterostomes. Protostomes literally means first mouth. This name refers to the fact that the blastula, the first cells to develop in the zygote, give rise to the mouth. In deuterostomes, the blastulopore gives rise to the anus. In some protostomes, a second opening results in the anus, and the reverse occurs in deuterostomes. Of the organisms we encounter in lab, sponges are the oldest animal forms. Sponges, Phylum Porifera, likely evolved from protozoans. Most are marine and live in a sessile existence attached to a substrate. They're non-motile, but their offspring are ciliated and motile. Sponges lack complex organization and therefore are asymmetrical and sometimes radially symmetrical. They have no germ layers, body cavities, or body plans. One advantage to this simplicity is that sponges can reproduce from small fragments of the organismal body that break off. There are three kinds of sponges categorized by the chemical elements that make up their skeletal elements, spicules. Chalk sponges are made of calcium carbonate spicules, glass sponges from siliceous spicules, and fibrous sponges from proteinaceous spicules. Cnidarians, phylum Cnidaria, include jellyfish, sea anemones, corals, and hydroids. Cnidarians have an ectoderm and an endoderm. The endoderm is also called a gastrovascular cavity because in this sac-like organism, the endoderm lines the gut. We referred to Cnidarians as having a tissue-level organization because there is no mesoderm that gives rise to the organs. Nonetheless, Cnidarians are motile and predatory. Their muscle tissue allows for movement and their nerve tissue allows for sensory communication and predatory feeding behaviors. Polyps attach to a substrate, and medusae are free-swimming cnidarians. Some cnidarians exhibit both life forms from an alternation of generations called metagenesis. The medusae generation produces sexually while the polyp form reproduces asexually by budding. Flatworms, phylum Platyhelminthes, are sac-like, bilaterally symmetrical, and hermaphroditic. They have three tissue layers but lack a true body cavity and therefore are acoelomates. There are two forms of flatworms, free-living and parasitic. Planarians are free-living and tapeworms and flukes are parasitic. With organ-level organization, flatworms have more complex sensory and digestive systems than cnidarians and sponges. Roundworms, phylum Nematoda, are bilaterally symmetrical, but unlike tapeworms, they are cylindrical, have three tissue layers, organ systems, and a false coelom. Like tapeworms, they can be free-living or parasitic. In this picture on the right, you can see an intestine that has been cut to reveal the lumen, which is filled with mature roundworms. Although I have not defined all these terms in this instructional video, you will become familiar with these vocabulary words as you work through the lab.